So as you've heard, OEHHA has a mandate for scientific review of food dyes in connection with their possible effects on children. And we'll be reviewing chemical, toxicological, biological properties of the food dyes in the course of this project, but I'm going to be focusing today on neurobehavioral effects of food dyes in animals. So these are the dyes we're going to be reviewing. They have many names. I'm going to be using the convention of the color and the number, red 40, yellow 5, and so forth. There's 7 of them, and they all have different things in common. They have to be reviewed individually from our perspective, but we do want to look at some of the things that they have in common. So these are the 3 azo dyes. They belong to a particular chemical group that has this azo bond in the middle of the molecule, good for producing color, and it's also a primary target for metabolism, so they can be metabolized similarly. These are the triphenylmethane dyes. They have this cluster of 3 aromatic rings around a carbon in their center, and so they also have some common properties. The other 2 dyes don't have anything structurally in common with the group. Blue 2 is a derivative of indigo, which is the dye that's used to color blue jeans. And red 3 is a derivative of fluorescein, which is a very widely used dye. So there's some common structural properties. Most dyes also have are sulfonated. They have a sulfur group here that promotes their solubility, and so that this is a place where chemical reactions can happen for most of them, all except erythrocin. But as chemicals, dyes also have things, these chemicals also have things in common because they're dyes. Some of the properties of a good dye include stability, so that the dye persists in the food processing and in food storage. They also have water solubility, so they're absorbed into foods and distribute throughout the foods. They have sort of indiscriminate protein binding, so that the color clings to proteins all throughout the food and provides a uniform color. And they have irreversible binding in organic materials, so that the color doesn't wash off or rub off. So another thing that we're looking at, in addition to some of the chemical properties, is the absorption, distribution, metabolism, and excretion of the dyes, how they enter the body, where they go, and how they're eliminated. That's represented by the ADME abbreviation. Some of the questions to ask: are there studies in people, and do people differ from laboratory animals in this regard? Are there studies in young animals and children, and do they differ from adults? And do we have the mathematical modeling that's commonly used to get a good, precise, quantitative measure of the ADME characteristics? There's also recent literature that's quite interesting. FDA is conducting studies of azo dye gut metabolism currently, looking at the genes that are involved and how they differ between species. There's some new information on triphenylmethane metabolism, and quite a bit of literature. We have quite a big folder on this topic: the protein binding, individual dyes, and how they bind to individual proteins, and what the biological implications are. Now, this may be a good time to point out that we're looking only in the animal studies at oral administration, and that in addition to foods, and as Dr. Mitchell will discuss, the dyes are contained also in medications and in supplements. There, of course, could be inhalation exposure and dermal exposure, because dyes are used in cosmetics, and they're also contained in vaping fluids. So our librarian, Nancy Furcho, conducted a literature review and came up with over 2,400 references that she's retrieved for us to look at. There are quite a few animal studies, in vivo and in vitro, and we found 31 with behavioral outcomes. Those are the ones I'll be discussing today. 15 of these with developmental exposures, 16 of these with adolescent adult exposures, and all of those exposures are in rodents, as represented by our little pictures of mice. Animal studies are very important because, as we've heard and we'll hear later from Dr. Taylor, I think, they are the basis of the safety assessment of food dyes. The dye manufacturers provide animal toxicology study data and production volume data and other exposure data to the FDA. The FDA then reviews that data to be sure that the dye isn't carcinogenic, very important because foods are covered by the Delaney Clause. And if not, they go ahead and find the NOEL, the no observable adverse effect level. That's the lowest dose in the toxicology studies that produces no toxic effect. Then they take the NOEL, according to the Red Book, they divide it by a safety factor or uncertainty factor, which has been mentioned, of 100 in order to get an acceptable daily intake for humans, the ADI. Then the ADI, as Dr. Lichtman said, is compared to an exposure estimate, the EDI, and it's determined that the dye is safe and can be certified. So this is all the basis, the beginning of all of this has to do with animal studies. Animal studies are very important. Dr. Voorhees and I enjoy reading them, and so we want to give you the results of what we've been reading about. So these are the number of studies with adult neurotoxicity and behavioral endpoints. The graph, the y-axis is the number of studies, and the different dyes are represented by their color. As you can see, red 3 and yellow 5 are the most studied, and there are no studies in the adult neurotoxicity literature of blue 2 and green 3. There are also mixture studies where the dyes are combined. And as you can see on the right side, there's quite a bit of recent activity in adult neurobehavioral toxicity testing of food dyes in the last couple of decades. So for developmental studies with behavioral endpoints, once again, red 3 and yellow 5 are the most studied. And Dr. Bradman will be explaining to you, I think, that from his work, that red 3 has the second highest consumption of all the food dyes in small children. The second most studied is tartrazine, tartrazine or yellow 5. This is the most commonly used dye in the food challenge studies in children. We do have one blue 2 study, but no green 3 studies as an individual dye, and quite a few mixture studies. As Dr. Voorhees pointed out, there are quite a few studies from the early times of the 1975 to 85, not so long ago for those of us that are retired. But there are also more recent studies that need to be folded together with the earlier studies in order to understand the developmental aspects. Now, as I mentioned, we're only looking at oral administration, but it's important to look at the way the oral administration occurs in the animal studies in terms of the relevance to children. So one approach is to put the dye into the diet of the animal. So the diet is ground up, the dye is added, the food is put in the food hopper, and the animal can eat at any time that it wants during the day and night. Then a daily dose is based on the food intake of the animal. So the animal actually determines what the dose is. This kind of administration is similar to the children's diet studies where you're comparing a diet with dyes to a diet with no dyes in order to determine what the effects on behavior are. Another common approach is the use of a single administration once a day, giving the entire daily dose at one time. This is similar to the challenge studies in children. It gives a higher dose and a higher exposure for the moment, but then the exposure during much of the rest of the day might be much lower. It kind of reminds us of the situation that was mentioned by several parents in the comments that they sent in on the symposium, that they wanted to understand what happened when their children were exposed to a lot of dye at a birthday party or during a snack attack or something like that, and then their behavior changed. Was that something real? Is that something we could find out more about? Another aspect is whether individual dyes or dye mixtures are used. In the human studies in children, both are used. In the animal studies, both are used. So when you're looking at the comparison of the studies, it's important to sort out whether you're looking at an individual dye or a dye mixture. Individual dyes are important because you can get causality when you give an individual dye. You know that that dye is causing the effect. Mixtures are important because they're much more common in the day-to-day life of children. So getting into some developmental study designs. And this slide has representatives of some of the studies from the developmental group. The dotted blue line represents exposure period, and the little red triangles represent testing times. So in this first study, the exposure to the dye began way back at the puberty of the parents, the FO generation. Both the males and the females, the parents were exposed right up to the time of breeding. Then the moms continued to be exposed through pregnancy and lactation. And when the little ones were weaned, they continued to be exposed through juvenile and adolescent development and up through young adulthood. You might ask yourself why anyone would do such a long exposure period. For those of us who are tox regulatory people, this is basically a lifetime study with an in utero component and a behavioral add-on that terminates a little bit early. So it's a common study design in toxicology. Other studies begin a little bit later, but continue for a long time, just before pregnancy, after pregnancy starts. There are some studies that expose only during pregnancy, and some studies that expose only during juvenile and adolescent brain development. So this entire period from the beginning of pregnancy all the way through puberty and beyond, now we know, is a time when brain development occurs and when it's appropriate to do a developmental toxicology study. Also, the behavioral assessments occur at a lot of different times. This is a time period shortly after birth when a lot of behavioral assessments occur in the studies. Also the juvenile adolescent period, and in young adulthood and even older adulthood, the behavioral studies may be conducted. So the overview from this slide is that there are a lot of different study designs. If you ask if something produces developmental toxicity, you can't really look for consistency across study designs. You have to look at each one of them individually and try to figure out what they're trying to tell you. Now to move on to some of the behavioral tests that are used, and Dr. Voorhees talked about this, but for the studies we're reviewing, these are the most common types of assessments. Behavioral development, looking at the animals shortly after birth for the maturation and appearance of simple behaviors in that time period. This is a picture of a riding reflex. Spontaneous motor activity is by far the most common behavioral test used. In this, the animal is put into arena and its activity is measured, but there are a lot of different ways to do that. Some of them are automated, so there's equipment that automatically measures the activity of the animals. Sometimes videotapes of the animals are reviewed and observers score their activity. The sessions can range from a few minutes, 5 minutes, to 24 hours a day for several weeks at a time. Once again, just to say plus or minus on activity isn't going to work. You have to look at the individual study to interpret the effect. And then finally, learning and memory studies. Learning and memory is an improvement in performance in a rewarded task with repetition. And the most commonly used tasks are water mazes, where the animal has to find a place to get out of the water by going through a maze. So I'm going to talk about one set of studies and one set of results because, as Dr. Voorhees says, it's important to really dig down into these studies to understand them. So this is a set of 6 studies that were done at the Tokyo Metropolitan Research Laboratory of Public Health. And there was one study on each of the 6 dyes, 6 of the 7 dyes that we're studying. It was a long-term exposure with the parents, both male and female, exposed before breeding, females continuing through the end of lactation, and then the offspring through the end of the study. These are behavior tests that were done. Behavioral development during the lactation period, And this is in the offspring, the F1 generation. The parents are called the FO generation. There was an activity test shortly after weaning during juvenile development, and another activity test after puberty in the young adult animals. Also, in these studies, they decided to measure the activity of the parents. So both of the parents were looked at before they were bred in the activity assessment. In these studies, there were 4 doses: control, low, medium, and high dose, a good dose response. And the authors were interested in comparing their doses to previous toxicity studies. So in most of the studies, they used the low dose for the study was 100 times the ADI, or the acceptable daily intake in humans. In some of the later studies, they used 10 times the ADI. And as you recall, 100 is the uncertainty factor, so 100 times the ADI would be the no effect level in the animal studies that were used to establish the ADI. Some results from this study. It's very important in these studies with long-term exposure, multiple life stages, to look at general toxicity. So these authors were good. They looked at mortality and growth throughout the study, and they reported no effects of the dye. They also looked at reproduction. There was some problems with pregnancy outcome, nothing statistically significant, but something to take into consideration. Then for the behavioral assessments, the pre-weaning development, no effects were reported. This is in the study of RED 3. Sorry, I forgot to mention that. For the maze learning, there were no effects. But for the activity evaluations at all 3 of the life stages that were studied in the dams pre-breeding and the offspring after weaning and the offspring as young adults, there were effects of activity at the high dose. Now to look a little bit more at how activity was measured, this is the arena that was used. It's a round pathway for the animals where they can move freely. And there are 36 photosensors around it, so every time the animal passes a photosensor, it makes a record of it, and this gives you a measure of activity. So the activity endpoints, and there's always a lot of them in an activity study: total number of movements or beam breaks, the total movement duration, total amount of time the animals kept moving, and then both vertical and horizontal endpoints. The number of vertical bouts. Vertical activity is the animal standing up on its hind legs. Since rats and mice don't walk on their hind legs, they immediately come back down. It's called rearing. That's vertical activity. Horizontal activity is running around the maze. And the endpoints for that included total distance, average distance of each bout of horizontal activity, average speed, and the number of turns. On the right side of the slide are some results from the first assessment, the assessment of the parents prior to breeding. The females in the FO generation, there was a significant effect on the number of turns in the maze. The high dose was lower than had more turns than the control group in this, and though it looks like there's something of a dose response trend, but the authors didn't analyze for trend. This is the second life stage when they measured activity. Their effects were found in the males, in the offspring at this time. It's hard to see. Let's see. This is the number of horizontal activity bouts. There were fewer of them in the male offspring in the high dose group than in the controls. However, the average distance for each bout was longer. The total distance was longer, but not significantly so. So more longer bouts of activity in the male offspring at this age. Oh, you actually can read that perhaps. Then at the third life stage in the young adults, there were effects in the female offspring. This is increased movement time, the number of time the animals kept breaking beams, increase in the total distance that they traveled during the session, an increase in the average speed during the session. Average distance was also greater than controls in the high dose group, but wasn't statistically significant. somewhat of an indication of a trend. No trend tests were done, and I should mention for this study, perhaps only for Dr. Voorhees' satisfaction, this was non-parametric analysis because of the variability in that mid-dose group. Now I want to talk about the relevance of the dose to humans, because if the study is nowhere near the human exposures, then it's a lot less relevant for the purposes of our project. So, as I said, the dye was administered in the diet, and this is the percent of diet that was used for the low, mid, and high dose. So this is a weight for weight, how much dye per total amount of diet. And this is the actual intake of the animals in milligrams per kilogram per day. This is the same measure that is used for the ADI. So acceptable daily intake is given in milligrams per kilogram per day. And so this is the amount that the animals ingested of the dye. Now you recall that the ADI is 1/100th of the NOEL, and conversely the NOEL that is used to produce the ADI is 100 times the ADI. So if you look at the ADI that they used to estimate the amount of dye to put in for the low dose, it was 0.1 milligrams per kilogram. So that would give an ADI NOEL, the NOEL that was used to produce it, of 10 milligrams per kilogram. These authors used the JECFA ADI. As Dr. Voorhees mentioned, JECFA is the World Health Organization Food Advisory Safety Committee, so they produce an ADI that's widely used in the animal experiments. And as you can see, that was a pretty good guess, because the low dose animals had 8 and 10 milligrams per kilogram. That's how much they ate, which is very close to what the investigators were estimating. Now, I think as Dr. Bradman will mention, the ADI for Red 3 is much different. It's the most different between JECFA and FDA. So the FDA ADI is 2.5 milligrams per kilogram per day. And that means that the NOEL that it was based on would be 250 milligrams per kilogram per day. Conversely, if you use the NOEL from this study, all the effects occurred at the high dose. So the mid dose was a NOEL. If you use 25 milligrams per kilogram per day as a NOEL to obtain an ADI, divided it by 100, you would get a value of 0.025 milligrams per kilogram per day. This is trying to get at the idea of whether neurotoxic endpoints or neurobehavioral endpoints are more sensitive than the endpoints that have been used previously to derive ADIs. Another developmental study design that's been used is the one where the dose is given only during pregnancy. This study used the JECFA ADI dose, not 100 times, but the actual ADI dose, and it gave a mixture of all 6 dyes. So this is a good mixture study, a mixture given during pregnancy. After the animals grew up, when they were adults, they were given the behavioral test, a Morris water maze test, a spontaneous motor test called the open field, and a forced swim test, and also some brain assays. So this is a good developmental toxicity study because it looks for permanent effects. They did find some effects in offspring a long time after the dose was given, which suggests permanent effects on the brain. But as Dr. Voorhees said, this is not really what we were looking for. We're not looking for long-term effects that are seen after an exposure. We're looking for effects that are seen shortly after an exposure, but can be reversed. So this study is not as relevant to our concerns as some of the other studies. These authors also did another study in which they upped the mixture, and they did find an increase in effects with 100 times the ADI dose of this mixture. Tells you a little bit about dose response. They also tested the offspring at an earlier age. so that they found the effects both in adults and in the offspring shortly after weaning. Now for a quick look at the adult neurotoxicity studies. We're calling it an adult study if the dosing started after puberty. The dosing durations here are usually about a month, so these are short-term studies. They use gavage dosing almost exclusively, giving 1 dose a day. And the behavioral evaluations are done during dosing, so that makes them more relevant to the situation in children. Most of them are hypothesis-based, which means the design is put together to try to answer a specific question. So this graph shows the doses in 11 of these studies, adult neurotoxicity studies with behavioral measures. Usually there's 3 doses. And you can see that the doses used vary a lot. So even with the same dye, yellow 5, a lot of different doses are used in the studies. And what we're seeing is that more recently, lower doses are being explored in these kinds of studies than have been explored in previous toxicity studies. Now, to give you an idea relative to human exposures, this horizontal black line is the ADI NOEL for that dye. So that's the NOEL that would have been used to derive the FDA ADI. In most cases, the studies, all the doses used are well below the ADI NOEL. There are some dyes in which the ADI NOEL is in the middle of the dose range of the study with neurobehavioral endpoints. So we're really looking at a different range of doses for these kinds of studies. Then this graph shows with the little arrow what the NOEL was in these adult neurotoxicity studies. Sometimes it was above the NOEL that was used for ADI development for these 2 dyes, but most of the times it was below, and sometimes it was quite far below. It's a little difficult to see these last studies, so here they are blown up, showing NOELs in adult neurotoxicity studies that are below 20 milligrams per kilogram per day, all of them, except this one. This one is not a NOEL because an effect was found at the lowest dose, so it's a NOEL. A low-L, we don't know what the NOEL is. Are you up for a little adult neurotoxicity data? So I just want to talk about 2 studies from one group that used a different design that I think is interesting from the viewpoint of the children's studies. So this is another Red 3 study. And in the first study, they gave 1 dose of dye, and then they measured activity every 30 minutes for 9 hours. So after they looked immediately after the dose was given. They had 4 doses, and a 5th dose was the control to compare the activity immediately after an exposure. So this would be more similar to the birthday party. But they reflected that this is not similar to what happens in children because these animals had never been exposed to dye before. So they did a second study with a pre-dosing period of either 15 or 30 days, and then they did the study with the single dose and measuring the activity afterwards. This is from their paper, and the first thing you'll notice is there was a decrease in activity after the dye was given. It was maximal at 2 hours after the dye was administered, and then it reverted to normal activity levels by 9 hours. In terms of dose response, these are the 2 highest doses down here. This is the 10 milligram per kilogram dose. At the 1 milligram per kilogram dose, there was no decrease in activity after the dye was given. So a dose-dependent decrease in activity with recovery at 9 hours. This is the second study. And the first thing you'll notice about this is there's an increase in activity instead of a decrease in activity. So, and it also peaks at 2 hours. It's somewhat dose dependent. There's a lot of different dose combinations here because you have both the pre-dosing period and the dose involved. But basically, the longest pre-dosing periods and the highest dose gave the greatest effect on activity. And once again, the activity returned to a normal level by the end of 9 hours. So that's the kind of study that maybe we'll look at more than people have before, because it's similar to the specific issue we're trying to understand, which is sort of the reversible effects of food dyes when they're given to children on behavior. Now, mechanistic data. As Dr. Lippman mentioned, causality is an important keyword in the FDA evaluation, and causality can really be boosted a lot by combining the information from the human studies, the clinical studies or population studies with the animal studies, and also with the mechanistic information. So we're reviewing that in some detail using some of the newer techniques, as Dr. Marty suggested. This is just a list of the proposed mechanisms that we're looking at. I could point out a few things. Neurotransmitters were looked at in the early days of the food dye literature. I think Dr. Voorhees did some of these studies, but back then we only knew about a few neurotransmitters. We have many more neurotransmitters and many more ideas about how they act. So looking at neurotransmitters is important. Probably the most common current mechanism under investigation in food dyes is oxidative damage due to reactive oxygen species that are produced with metabolism of the dyes. And then there's the issue of inflammation, hypersensitivity, and histamine production with dyes, a very interesting area in which there's quite a bit of in vitro data that could be folded into the human studies. Evaluation of the studies. So this is what we're currently doing. It's a complex set of studies, and we need to evaluate it correctly. We're summarizing all the studies in tables using the ARRIVE guideline categories. These are guidelines established by this National Center for Replacement, Refinement, and Reduction of Animals in Research to determine whether studies are fully enough reported to be evaluated with some certainty. We're adding to the ARRIVE guideline categories whether or not the statistics were litter-based, an important consideration in our area, and whether there was a background of general toxicity. Then once we understand that we can evaluate the studies confidently, there's enough information, we'll be looking at study power and sensitivity of the measures, the consistency of the findings, dose-response relationships, and possibly keeping an eye out for data sets that are appropriate for risk assessment. So our conclusions at this point: review of neurotoxicology studies adds a new dimension to understanding the toxicology of food dyes. Comparisons of human exposure data to doses used in animal studies helps identify the relevance of the animal studies to food safety. And the animal studies must be integrated with mechanism studies to help evaluate the biological possibility and to support causality in human studies. I need to acknowledge the people that helped me put this information together and prepare the presentation: Anna Smith, Marjani Akindunde, Mayela Gian, and Ruan Tilkaratni. And on some of the slides, there were many references for the data that was presented. So we're providing a reference slide that gives you the full reference for any of the data slides. Thank you. I don't know if we have time for questions. It's almost break time.